Today we are going to be learning about enzymes. This portion is taken from a book publication called Pathfinders. This is a very nice book. So let's start. An enzyme is a biocatalyst that increases the rate of a chemical reaction without itself being changed in the overall process. Virtually all cellular reactions or processes are mediated by enzymes. Enzymes have several properties that make them unique. Number one, let's see, most but not all enzymes are proteins. This is a good point to remember. Wait a second. They are all proteins, with the exception of a small group of catalytic RNA molecules, and all enzymes are proteins. Enzymes are highly specific. They are specialized proteins and have high degree of specificity for their substrates. Enzymes exhibit enormous catalytic power. It increases the rate of reaction by lowering the activation energy. Enzymes do not change equilibrium state of a biochemical reaction. It changes only the rate at which the equilibrium is achieved. Proteinaceous enzymes can be divided into two general classes. Number one, simple enzymes consists entirely of amino acids, and conjugated enzymes consists of protein as well as non-protein components. The non-protein components is called a cofactor. This conjugate we are talking about right now, this conjugated enzyme, this consists of protein and non-proteinaceous component. Non-proteinaceous component is called cofactor, and it is required for catalytic activity. Removal of cofactor from the conjugated enzyme leaves only protein components called the apoenzyme, which generally is biologically inactive. The complete biologically active conjugated enzyme, simple enzyme plus cofactor, is called holoenzyme. So let's go through this again. Proteinaceous enzymes, there are of two types: simple enzyme and conjugated enzymes. Simple enzymes are entirely made up of amino acids, and the conjugated enzymes are made up of two components: protein component and the non-protein component. The non-protein component is also called a cofactor. This requires a catalytic activity. Removal of this cofactor from the conjugate enzyme leaves the proteinaceous part, protein component, and this protein component is called apoenzyme. It is generally inactive in nature. So a complete biologically active so what are the biologically active components in the whole proteinaceous enzymes? They are this amino acids from the simple enzymes and the non-protein portion, that is called the cofactor. So if we put this simple enzyme plus cofactor, it is called the holoenzyme. So we did this till here. Now we'll move forward. A cofactor can be linked to a protein portion of the enzyme either covalently or non-covalently. A cofactor can be linked to the protein portion either of the enzyme either covalently or non-covalently. Okay, some cofactors are simple metal ions. Other cofactors are complex organic compounds, which are called coenzymes. Many coenzymes are vitamins or contain vitamins as a part of their structure. Some coenzymes are only transiently associated with a given enzyme molecule so that they function as co-substrates. Enzymes which are tightly associated with protein covalently or non-covalently are called prosthetic group. Enzymes that depend on metal ions as cofactors fall into two categories. Metal activated enzyme bind metal ions weakly during the catalysis, and metalloenzymes. Enzymes binds the metal ion very tightly or require the metal ions to maintain its stable native state. So enzymes are mostly proteinaceous. Some are made up of RNA. They are called ribozymes. The proteinaceous enzymes are of two types. Simple enzyme, they are made up of amino acids, and conjugated enzymes, they have two components, a non-proteinaceous component and a proteinaceous component. The non-proteinaceous component is called cofactors and proteinaceous component is called apoenzymes. The non-proteinaceous component are of two types, one which has metal ions and the other which has organic component, and it is called coenzyme. So in coenzymes there are prosthetic groups and co-substrate. Prosthetic groups are tightly bound. They are tightly bound with the protein. They are covalently or non-covalently, but the ones that are loosely bound are called co-substrates. They are only transiently associated with the given enzyme molecule so that they function as co-substrate. The metal ions are either metal activated enzymes or metalloenzymes. The metal activated enzymes are the enzymes that binds metal ions weakly during catalysis, and the metalloenzymes are the one they bind very tightly or they require the metal ions to maintain a stable. So this is also tightly or weakly. Simple difference between these both of them. Let's move forward. So we just read the coenzymes are vitamins, or they contain vitamins as a part of their structure. Okay, so some vitamin B complexes and their coenzyme forms we can see. So thymine, that is B1, they form thymine for a pyrophosphate. The reaction or process promoted, they promote or in a part of reaction of decarboxylation, aldehyde group transfer. Second is riboflavin B2. Coenzyme, they form FAD, FMN, redox reaction. Pyridoxine B6, pyridoxal phosphate, amino group transfer. Nicotinic acid, niacin, NADH, NADPA, positive for the redox reaction. Pantothenic acid B5, coenzyme A, acyl group transfer. Biotin, biocytin, carboxylation. Folic acid, tetrahydrofolic acid, 1 carbon group transfer. Vitamin B12, deoxyadenosyl-cobalamin, intramolecular rearrangement. These are the coenzyme. You see these coenzyme forms. We will be reading about these in the further portion of this metabolism portion, like in some cycles of glycolysis or fatty acid transfer, or these are the redox, this acyl group transfer coenzyme A is used in from pyruvate to TCA cycle conversion. It is used there. I think it forms from pyruvate to acetyl CoA. So that CoA is this coenzyme A. Vitamins in human diet that are coenzyme precursor are all water soluble vitamins. So now we will be thinking about how are they named, and what are the classifications of these enzymes. Many enzymes have trivial names. Some enzymes are named by adding suffix ase to their name of the substrate upon which they reacted, as in urease for the urea hydrolyzing enzyme. Other enzymes require name bearing little resemblance to their activity. For example, trypsin, a proteolytic enzyme, is secreted by the pancreas. Trivial names provide little information about their reaction that the enzyme catalyzes. Because of the confusion that arose from these trivial names, the Enzyme Commission, now we have an Enzyme Commission too, EC, has given all known enzymes a systematic name in addition to its existing trivial name. Systematic names consist of two parts. The first part of the name, contains the name of the substrate or the substrates separated by a colon. Second part, ending in ase, indicates that the nature of the reaction. For example, malate dehydrogenase. Trivial name, interconverts L-malate or oxaloacetate using NAD+ as a coenzyme. Wow. Its systematic name is L-malate NAD+ oxidoreductase. So there was this enzyme malate dehydrogenase. This was the trivial name. What does it do? It interconverts L-malate and oxaloacetate using NAD+ as coenzyme. Okay. So this goes to this interconversion na, and this goes to this. Simple. It uses NAD+. So what were we doing? So it should have, the systematic name should have two parts. The first part should contain the name of the substrate or substrates. So the name of the substrate, L-malate, okay, substrate separated by a colon. Okay, there is a colon because it is using NAD+, so we have NAD+ here. And the second part in ase, ending in ase, indicating the reaction. What kind of reaction is this? This is oxidoreduction. Okay, so oxidoreductase. The Enzyme Commission has classified enzymes into 6 main classes on the basis of the reaction catalyze. Within each classes are subclasses, and under each subclasses are sub-subclasses within which individual enzymes are listed. According to the system, the Enzyme Commission has given each enzyme a number termed EC number for 4 parts, like EC 2.7.1.2. EC number for enzyme hexokinase. The first 3 numbers define class. subclass and sub-subclass respectively. First three numbers: class, subclass, sub-subclass respectively. The last number of the serial is subclass, indicating the order in which each enzyme is added to the list. Thus, the EC number is a numerical naming scheme for enzymes, which in strict sense do not specify enzymes, but enzyme catalyzed reaction. Six enzyme classes are EC1, oxidoreductase. What happens in oxidoreductase? Oxidation and reduction reaction. A is oxidized, B is reduced. Transferase, transfer of an atom or group between two molecules. AB and C is present. B goes from A to C. The transfer of an atom or a group. Transferase. Hydrolase, hydrolysis reaction, water is added. So when water is added, H comes here and OH comes to the other one. Lyase, removal of a group from substrate, not by hydrolysis. This is removal of the group. Water is not added, but the removal is group is happening. So there was this double bonded guy. HX is coming. Okay, this HX, X is going to one person and B is going to the H. Okay, lyase. Isomerases, isomerization occurs. So it looked like this, now it is looking like this. Change in configuration. Ligases, what happens? Ligase joins, catalyze synthetic reaction coupled with hydrolysis of nucleoside triphosphate. Okay, so energy is added in this. A plus B plus ATP forms AB plus ADP. Ligase is the recommended name of synthase. Biochemical nomenclature distinguish synthetases and synthases. What is synthetases and synthases? Synthases catalyze condensation, reaction with no nucleoside triphosphate. Okay, in which no nucleoside triphosphate is required as an energy source, but in synthetases catalyze condensation that do use nucleoside triphosphate as an energy source for this synthetic reaction. A synthase is a lyase and does not require any energy, whereas synthetase is a ligase, thus require energy. Okay, so did we learn about lyase? Yes, lyase. Removal of a group from a substrate, not by hydrolysis. Condensation, no required, whatever. 6 enzyme classes have been recognized since the first enzyme classification list was approved by the International Union of Biochemistry in 1961. These classes are oxidoreductase, transferases, hydrolases, lyases, isomerases, and ligases. However, it has become apparent that none of these could be describe the important group of enzyme that catalyzes the movement of ions or molecules across the membrane or their separation within the membrane. These enzymes have now been classified under the new EC class of translocases. Okay, so there is a 7th class translocases. Very important, very important to remember. The 7th class translocases. What happens in translocations? There are the enzymes, group of enzymes that catalyze the movement of ions or molecules across the membrane, or their separation within the membrane. These enzymes have been classified under the new EC class of translocases. The reaction catalyzed are designated to transfer from side 1 to side 2. Meaning of EC number. Meaning of EC number. The first digit in the EC number indicates the oxidoreductase, this EC1. Okay. The enzymes belong to this class catalyze the oxidation reduction reaction. The second digit, the subclass, this in the EC indicates the donor of the reduction equivalent, the hydrogen or electrons involved in the reaction. The third digit, subclass, refers to the hydrogen or electron acceptor. Class 1, oxidation reduction, subclass 1, 2, 3, 4. Subclasses 1 is alcohol, aldehyde ketone, then 3 is primary amine, 4 is secondary amine. The subclass hydrogen or electron acceptor, who is accepting? 1 is NAD+, 2 is Fe3+, 3 is O2. The fourth digit, the order in which each enzyme is added to the list. Okay, understood? Trivial names of oxidoreductases include oxidases, use oxygen as an electron acceptor but do not incorporate into the substrate. Dehydrogenases use molecules other than oxygen. Example, NAD+ as an electron acceptor, and oxygenases directly incorporate oxygen into the substrate, and peroxidases use H2O2 as the electron acceptor. However, the first digit of the EC number, EC5114, indicates the isomerases, EC5, the first digit. Enzyme belong to this class catalyzes the isomerization reaction. The second digit, the subclass of the EC number, indicates the type of reaction. Okay. And the third digit refers to the type of molecule undergoing the reaction. Okay. So in isomerization reaction, its subclass shows the type of reaction. So number 1 is racemization of epimerization. Okay, isomerization. What is isomerization? Change in the structure. So there are different ways in which this change in structure can take place. Like number 1 is racemization or epimerization. Number 2 is cis trans isomerization. There are other isomerizations also that is not named here. So this subclass gives the substrate, which substrate is growing from here to there. So for one is amino acids, other is hydroxy acids, and the fourth digit, like the previous one, it gives the order in which each enzyme is added to the list. So how enzymes operate? An enzyme accelerates, wait a second, I'm sorry. Yeah, so an enzyme accelerates the rate of a chemical reaction several times as compared to the uncatalyzed reaction in the water. It increases the rate of the chemical reaction by lowering the activation energy. However, an enzyme does not change the free energies of the substrate and the product. Very nice. The free energy of the reaction, delta G, remains unchanged in the presence of an enzyme. So the relative amount of reactants and the products at the equilibrium are unchanged. In other words, an enzyme does not change the position of equilibrium in a chemical reaction. It only accelerates the attainment of equilibria, but do not shift their position. The formation of an enzyme substrate complex in the first step is enzymatic catalysis. The region which contains the binding and catalytic sites is termed the active site of the enzyme. The unique physical and chemical properties of the active site limit the enzyme activity of the specific substrate. The substrate binds to the active site of the enzyme by multiple weak non-covalent interactions like hydrophobic interaction, H-bond, ionic interaction, or reversible covalent bonds. The free energy released in the formation of the large number of weak interactions between the enzyme and the substrate is termed the binding energy. The binding between the enzyme and the substrate is highly specific. A given enzyme usually binds only to one kind of substrate. The specificity of the binding depends on the precisely defined arrangement of the molecules, arrangement of the atoms in the active site. The specificity of the enzyme substrate interaction arises mainly from the hydrogen bonding and the shape of the active site, which rejects the molecules that do not have sufficiently complementary shape. Two important models have been proposed to describe the binding process between the substrate and enzyme. The first is lock and key model, assumes a high degree of complementarity between the shape of the substrate and the geometry of the binding site on the enzyme. The complementarity between the enzymes and their substrate is the basis of lock and key model of the enzyme function. This was first proposed by Emily Fisher in 1894. So we can see in this, the lock and key model, they should be complementary to each other, otherwise they will not fit. Okay, so the enzyme is here, enzyme and goes to the substrate, it makes an enzyme substrate complex. The enzyme remains unchanged, but the product is formed. So the enzyme leaves it, stays unchanged, and the product is formed in a much higher rate. According to the induced fit model, the second model we are talking about, induced fit model given by Daniel Koshland in 1958, about 60 years later. Wow, 62 years. The enzymes are flexible and that the shape of the activity site can be markedly modified by the binding of the substrate. The binding of the substrate induces the conformational change in the enzyme that results in a complementary fit once the substrate is bound. The binding site has different three-dimensional shape before the substrate is bound. So there's this shape. The substrate makes enzyme substrate complex, it remains unchanged and the product is formed. So induced model, the enzymes induce the shape according to the substrate. Induced fit model, in this model the enzyme changes shape of substrate binding. As soon as the substrate binding occurs, enzyme changes its shape. The active site forms a shape complementary to the substrate only after the substrate has been bound. Transition state theory and activation energy. In both uncatalyzed and enzyme catalyzed chemical reactions, substrate changes into the product goes through a transition state. The transition state is the intermediary state of the reaction where the molecule is neither a substrate or a product. It is unstable state and will very quickly break down to form the product or back to the substrates. It is the point of the highest free energy in which the substrates are partially converted to the product. The difference in free energy between the transition state and substrate is called gives free energy of activation or simply activation energy, symbolized by delta G and this sign plus plus. The greater the value of activation energy, the slower the reaction. Enzymes lower the activation energy, but where does the energy to lower the activation energy comes from? Lowering the activation energy occurs due to the binding energy. The binding energy released as a result of the interaction between the energy and the substrate. So, an enzyme and the substrate. Only the correct substrate can form maximum interaction with the enzymes. Only the correct substrate can form maximum interactions with the enzyme and thus maximize the binding energy. Furthermore, the full complement of such interaction is formed only when the substrate is converted into transition state. Thus, the maximal binding energy is released when the enzyme facilitates the formation of the transition state. The combination of substrate and enzyme creates a reaction pathway whose transition state energy is lower than that of the reaction in the absence of enzyme. Because of the activation energy is lower, more molecules have energy required to reach the transition state. Decreasing the activation barrier is analogous to lowering the height of a high jump bar. More athletes will be able to clear the bar. It was here, now it is here. So obviously more will be going. Okay. So when only the uncatalyzed reaction, simply from substrate to product, substrate is there is an high activation energy, it is going there and coming down, gives free energy for the reaction, product is formed. Now in the enzyme catalyzed reaction, substrate plus enzyme first is doing and then product plus enzyme they are doing. So substrate plus enzyme, they are right now not bound. So when they are bound, we see the activation energy is very low. Okay, where does this energy go? To the binding energy. And now the binding opens up and we get a product and the enzyme. Nothing happens to the enzyme. The figure of free energy changes for a spontaneous reaction proceeding via the formation of transition state. An uncatalyzed and enzyme catalyzed chemical reaction in which the substrate S changes into the product P goes to the transition state S star P. The point of the highest and free energy is the transition state in which the substrates are partially converted to products. Activation energy is the difference in the free energy between the substrate and the transition state. An enzyme cannot alter free enzyme or substrate and product. It can only decrease activation energy to allow the reaction to approach equilibrium, where the rates of the forward and reverse reaction are equal. More quickly, then it would be absence of the enzyme. So we are doing till here today. More in the next video when I will be up to do this. So till then, have a very nice 24 hours. Be happy. Good night. Goodbye.